Hello grade 12! So we're carrying on with part 2 now of dealing with autosomal monohybrid crosses. So make sure that you watch part 1 before we go into part 2. So as a reminder, what we did in the previous video, I went through the question relating to if a purebred black rabbit had offspring with a purebred white rabbit, and what would the results be. And we found that through our Punnett square that we were then going to get all heterozygous black furred rabbits. So in that instance, we let capital B represent black fur, which was the dominant trait, and little b represent white fur, which was the recessive trait. So now what I'm going to do in this video is I'm going to change up the question slightly, and I'm going to dictate for you, I'm going to give you the step-by-step process that you need to follow to answer a question that is Punnett square based, or monohybrid cross based. So I am now going to give you scenario 2. So I'm going to say the filial 1 generation black rabbits, the heterozygous black rabbits in the previous question, were bred together. What will the potential genotype and phenotype ratios be? So I've dictated what they need to be here. So we are going to use the same assumption that we did previously. We're going to let capital B represent black fur and little b represent white fur. So this is the steps that you need to follow in order to answer a Punnett square question. So please write these down as we go. Learn these steps to know that this is what I need to do. Make sure you get everything, because you'll see where the marks for these kind of questions comes in. So step 1, you must identify the parent genotypes. Sometimes we are very nice and we give these parent genotypes to you in the question, but otherwise you must, part of the skill of this question is, can you identify what the genotypes are of the parents? Even if it is given to you, you must state the parent genotypes that you are using, because it is possible for someone to make a mistake, it is that someone didn't read correctly, so it is still part of the skill. So in our example here, remember I told you the F1 generation, the heterozygous black rabbits. So in this example, the parents are going to be both of them big B little b crossed with big B little b, because both of them are the same. All the F1 generations were all big B little b. So again, this is a follow-on from the previous question we did. If it was a standalone question, you would probably be given a bit more information to allow you to identify this. But this is what we know. And that is step 1, identify the parent genotypes. So these are heterozygous parents that we know. Step 2, draw the Punnett square and where the alleles are going to be, or the different versions of the gene. So from our example, you physically draw the actual Punnett square, make sure it's big enough, don't do it small, make it big, like really don't scrimp on the space here. And then in this example, we are going to show that the one set of gametes, or the one set of genes, the one set of alleles is on this side, one big B, one little b, and the other set on the other side, which will be the same, because they are both big B, little b. So that is step 2. Step 3, fill in the spaces correctly, as I showed you. Now it's going to be slightly different, but again, you follow on. As this one joins into this square, what's going to join on from the top square? So in this example, in this square, it will be big B, because that is the combination in this square. If I go move along, it will be big B, little b. If I come down to this square, it will be big B, little b. So we always try and put the capital letter first, if it's heterozygous as well. So you could say little b, big B, but put the capital letter first as a sort of semantic version. And in the last square, it's going to be little b, because those two have combined in that square. Okay, so now we've drawn the Punnett, and we filled it in. Now the rest of the question is going to depend on the requirements that have been asked for you. So in my question that I asked you, I asked what are the potential genotype and phenotype ratios. So I'm focusing on the ratios. I didn't ask you what are the potential genotypes. That's a different thing. So I'm following on as we would go from the next thing. So depending on what is required from you, you could ask me to do step 4, identify the genotypes and or the genotype ratio. So in our example here, the genotypes are big B, or it will be big B, little b, or it will be little b, little b. So I separate these with a semicolon. That's important. It's a semicolon that I'm separating, or a comma, you could say, I suppose. But those are only the potentials. I'm not saying 2, but these are the potential combinations I can have. Now, what is the genotype ratio? The ratio is the representative prediction. So for this example, it will be 1 big B, colon, is to 2 big B, little b, is to, colon, 1 little b, little b. So I literally counted how many times each of these will occur within this. It's like another way of saying there's a 25% chance that it's big B, because it's separated into 4, a 50% chance that it's big B, little b, and a 25% chance that it's big B, little b, little b. But I always do my ratio in the smallest ratio, smallest denominator possible. So if it's 25, 50, 25, that is a 1 to 2 to 1. So that is genotype, step 4. Step 5 is then identify the phenotypes and or the phenotype ratio. So remember the phenotype, the outward physical expression. So what's going to happen here in our example? Well, our phenotypes are going to be 3 black and 1 white. Slightly confused? Remember, there is a difference between genotype and phenotype. So as much as in the previous question I had the potential of a big B, and a big B, little b, the phenotypes of those ones are still going to be black. So whether it's homozygous dominant or heterozygous, they will still be black. So I don't say 1 purebred black, 2 heterozygous black. No, it is 3 black, and then the 1 which is the little b, which will be white. So remember, it can only be white if it is both recessive genes. So 3 black to 1 white. What is the phenotype ratio? The only difference is I add a colon. 3 black to 1 white. So it's like saying you have a 75% chance that it's going to be black fur and a 25% chance that it will be white fur. Lowest common denominator then, 3 to 1. Okay, so don't get confused when we say what is the phenotypes, they are black and white, versus the genotypes where it could be homozygous and heterozygous. So in this instance, you would get 3 black and 1 white. Okay, so this is your summary. These are your 6 steps. Step 1, identify the parent genotypes. Step 2, draw the Punnett square and the alleles. Step 3, fill in the spaces correctly in the Punnett square. Step 4, identify the genotypes and the ratio, if the question asks it, whichever it asks. Step 5, identify the phenotypes and or the ratio, if the question asks it. And then I'm adding in a step 6, which is based around a probability question. And I'm going to do that in the next example you'll do by yourself. Sometimes you are asked, and they'll say, what are the chances that the offspring will be, let's say in the previous one, will be white? So if I was looking at the previous example of the heterozygous combination, what is the probability that it will be white? Well, there is a 25% chance that it will be white. What is the probability that it will be black? A 75% probability that it will be a black fur. So pause the video, write down those if you haven't done that already. But otherwise, I'm then going to pose to you a practice question. So you're going to pause the video, you're going to do this by yourself as the 6 steps that I've told you to do now, and then we'll check your answer. So I pose to you a heterozygous brown-eyed individual, something different here, has offspring with a homozygous blue-eyed individual. What are the genotype and phenotype ratios? And what percentage chance will their offspring have blue eyes? Yes, you have all the information that you need here. It'll take a little bit of thinking. Do all your 6 steps. Write it all down as if it would be an answer. Pause the video. Do it now. Okay. So let's go through the answer. I really hope that you've done that because this is a very tricky question that we've got. So yes, I do have all the information that you need here. I'm going to go through it. So your first step is to assign letters to the different genes. So what am I looking at? Eye color. I've got brown eyes and blue eyes. So I have said here, let brown eyes equal the letter A, and let blue eyes equal little letter a. So again, it doesn't matter. I don't have to use B, don't have to use E. I've just assigned a letter. So I'm just showing you. If you used a different letter, that's fine, just as long as they equate to the capital and the lowercase. So now why have I let brown eyes be the capital and blue eyes be the lowercase? So the parents, step 1. The person with the brown eyes is capital A, little a. And the person with the blue eyes is little a, little a. How did I get that? Well, remember, I've got heterozygous brown and homozygous blue. Therefore, if it's homozygous blue, but in the heterozygous form it is brown, clearly brown then is the dominant color. Remember your definitions of what is a dominant and a recessive? The dominant can be expressed both homozygously and heterozygously, but the recessive gene can only be expressed as homozygous. So the mere fact that I've got the heterozygous brown clearly means that whatever that color is shown is the dominant color because we are dealing with a heterozygous. Okay, so I hope you didn't get tripped up with that and that you could have figured that out. If you haven't and you got stuck at that point, pause, carry on from this point here. But I'm going to carry on. So that was step 1, do the parents. Step 2, draw the Punnett square. These would be my allele things. Again, if you switch these the other way around, it doesn't matter as long as they are the correct sides for each. If I was to fill these in, these would be what I would fill them in as. Check that you got the correct combinations. Okay, then first question, what will the genotype ratio be? The genotype ratio is 1 big A, little a, is to 1 small a, small a. Why is it not 2 to 2? Because remember, we always have to make it as the lowest common denominator. So 2 to 2 should be written as 1 is to 1, because you are essentially saying there's a 50% chance, 50% chance. For every 1 a, big A, little a, there is 1 little a, little a. Okay, next one. What is the phenotype ratio? Again, same thing. Lowest common denominator. 1 brown-eyed is to 1 blue-eyed. So again, we've got the 2 heterozygous here, which means they will be brown-eyed, and the 2 homozygous, which means they will be blue-eyed. So that is your phenotype, lowest common denominator. 2 to 2, but it's still 1 to 1. And then the final question I asked is, what is the percentage chance that they will have blue eyes? And it is a 50% chance that they will have blue eyes. So that would be the whole answer for this question. You would have needed to have all of this and done correctly for you to get the marks. And this is where the marks would have gone, just as a matter of interest. So, 2 marks that you correctly identified each parent's genotype. 2 marks that you actually filled in the Punnett square correctly. You didn't make a mistake when you were assigning these. 1 mark to the genotype ratio. 1 mark to the phenotype ratio done correctly, and 1 mark to the percentage chance. So that is 7 marks. That's a big question. But really, if you know what you're doing, it really isn't that difficult. Again, the most difficult part was identifying the parent genotype, but we had all the clues and the information there for us. The rest was fairly easy. So if you didn't get that right, please go back and do that, because this is a very easy example that we do. When we get to sex-linked, it becomes a lot more difficult. So please make sure you understand this. Please make sure you get this. Please make sure you know how to identify from an autosomal perspective how to identify parent genotypes and the rest of the steps from there. So we're going to move on to something different after this. Might give you a few practice examples, but otherwise, please go through this. Make sure you're understanding things. Go through the terminology videos again if you need to. But otherwise, I hope you're understanding. I hope you're getting this. Make sure you go through things. Happy studying. Keep up the hard work, guys. Hope you're doing really well.